Okay, thank you, Tom, and thank you to all of you. It is a privilege to be here and speak before you. And I want to make it interactive. I'm going to ask you some questions during this, and I want you to shout out or raise your hand, whatever you want to do. All right, so this morning we've heard about the devastation of the 1918 influenza virus, and also that our seasonal epidemics can be just as concerning. But, in the last 5 years, there's been several other public health incidents or outbreaks that have garnered a lot of both media coverage as well as funding, including it was only 5 years ago that we had Ebola in the United States. More recently, we had Zika virus. And people think about that just being a problem in Latin America, but this map, which you can't read, but the blue, that's okay. The blue, just go for the blue colors, and it shows you the number of symptomatic Zika cases that actually happened here in the United States, including several in Pennsylvania. And now we're dealing with something else. Who knows what this is? Measles, exactly. And this is where we're at with the measles outbreak. 1,250 cases, 10% of those hospitalized, 31 states. So there's one state on this map that is not dark blue, meaning it has measles cases. Anybody want to guess what that is? Hawaii. So we know this is from people not being vaccinated. And when you get the measles vaccine, that's not the only virus you're vaccinated against. There's other things. So what's the new outbreak that we're facing? Mumps. Exactly. Very good. And so in spite of all this, there's some, these are big problems, but there's also features that make influenza unique as compared to these and a bit harder actually to control. And one thing that we've heard about this morning is the fact that they're zoonotic. There's an animal reservoir. We can get infected from animal influenza viruses, including in some cases your dogs and your cats. Now, flu is by no means the only zoonotic virus. We have MERS, SARS, Nipah, and Hendra. And the key is that all of these have a host species or a reservoir where the virus actually maintains itself in the environment. And as we've heard, that's in birds in the case of influenza virus. Now, this is what we consider our wheel of influenza misfortune. And you can see in the middle that there's a duck. And that's because ducks and wild waterfowl are actually the reservoir for influenza viruses. They have many strains that we don't see in humans, at least not yet. And what's unique about the duck is they can get infected with even very pathogenic strains of the virus, and it doesn't make them sick. And also, in many cases, it's not a respiratory illness. It comes out the other end of the bird. And so what happens when birds migrate and they fly over your head? What do they like to do? Poop. Exactly. They poop. And so that's what they do. You have these influenza-infected birds releasing virus into the environment and gets into our marine mammals, can get into horses, poultry, pigs, and they can establish new lineages in these animals. For example, a bird virus got into horses, established a new lineage that now circulates in horses, and has crossed into dogs. What we don't typically see, at least to my knowledge, is any of these wild waterfowl viruses getting directly into humans. They typically have to go through these domestic poultry. And these are these cases of bird flu that you've heard about this morning and you've heard about in the news. And these really started in 1997 in Hong Kong, where 5 people got infected with highly pathogenic avian influenza, H5 viruses, for the first time. It sort of stopped once they cleaned out those live bird markets, but then it came back again in 2001. And since then, there have been people infected with H5 influenza viruses around the world. Now, as we've heard, these are very severe viruses, high mortality, pneumonia, looks a little bit like the 1918 flu, but they don't transmit well. So we're a dead-end host for them. The virus infects that person, but it won't go to other people. But H5s aren't the only ones that we're concerned about. More recently, we had H7N9 outbreak in China, and this was actually, watching as a flu person, a bit of a scarier situation because the chickens that were infecting people were not sick. So we couldn't see it in the environment, we couldn't see it in these markets, and so they weren't getting rid of it in the environment. And so you can see what happened where we had 5 waves and many more cases of human flu, but again, very limited transmission. And what's occurring now out in nature is basically, if anybody had their chicken soup, it's kind of alphabet soup, where there is a number of different strains of avian influenza viruses crossing in people, sort of all the time. But again, with very limited to no human-to-human transmission. What we really worry about are these interfaces. The situations where these wild birds are in contact with poultry, domestic ducks, and they're in contact with mammals. Where does this happen? Places like live bird markets, which are in cities all around the world. It's a cultural thing to go pick out your fresh meat for the day, and you have these asymptomatic ducks potentially shedding virus to these chickens, which may or may not get sick and expose the mammals. This is happening all around the world in backyard systems, right? More people are keeping backyard chickens, backyard pigs. These are great areas where influenza can transmit from wild birds, look at these little ducks, to chickens, to pigs. And our concern is when this comes into play, right? The people that are in contact with that. So this is another unique feature of influenza virus and why we worry about these interfaces. And it was introduced to you this morning, but I'm going to go into a bit more detail. And this is that influenza viruses can undergo significant genetic variation through 2 ways, antigenic shift and drift. Let's talk about shift first. So here's the influenza virus. It has 2 surface proteins that are on its head, hemagglutinin and neuraminidase. This is what you want to make antibodies to. And then, as you heard from John, all of its genetic components are in individual segments, right? So think about this. What we're concerned about is you get 2 viruses going through a mammal, a bird, whatever, and actually being able to exchange these segments, and something new comes out that can potentially go back and infect these species. So you think about it like having 8 red and 8 green M&Ms. Put them in a pile, grab handfuls of 8. Where's my mathematics people? How many different combinations would you get from something like that? And this is what's happening out in nature all the time, and as you've heard, this is what causes pandemics. And here's the genetic composition of the pandemics that we've had. Here's the H1N1 that, as you heard from Jeff, appears to be a wholly avian virus. In contrast, your H2N2 was a reassortment between avian and human, and again, we assume it went through some kind of mixing vessel, a pig, but it may not just be pigs. That's always a paradigm, but perhaps people could be a mixing vessel. H3N2 had your H3 avian, some H2 human, and the 2009 pandemic was something that we probably could never have dreamed up even in the laboratory, how genetically complex this virus was. So if you look at it, this was actually 4 separate genetic introductions into pigs. It really started with the pigs in the late 1990s in the Midwestern US. So it took years for this virus to actually evolve. But what happened is you've got pigs that got infected with classical swine influenza viruses. And these, we talked about the H1N1 from 1918 persisting in people. Same thing happened in pigs. Pigs got infected with the 1918 viruses, and that lineage has continued throughout the pig population. So you've got 1918 or classic swine genes, a North American avian, which was a turkey from Minnesota, a human H3N2 virus, and I'm losing my pointer here, and then we don't know how, but a Eurasian avian-like swine virus, all mixed in pigs and ended up being the 2009 pandemic virus. So there's a lot of influenza virus in animals around the world. So here's the known outbreaks of avian influenza in poultry. See, look here in the US. And this is somewhat deceiving in that the only thing reported here are the pathogenic H5s and H7s. So we don't even know about the H9s, the bird H1s. It's going to be much, much more than this. So take a look at pigs. Here is the pig outbreaks all around the world. And there's a really interesting, if you overlap these, take a look at this. China, Europe, US. So you see there's a lot of different opportunities for these viruses to cross. And Jeff showed you a complex sort of lineage of human influenza viruses. Look what's happening in the pig populations, right? Is starting in 1979, we got another emergence, to what we have circulating today. And it's different in the US than it is in Europe. And again, remember, these pigs, for the most part, are asymptomatic. Most of the birds are asymptomatic. Makes it difficult. And there are areas of the world where we really have no information, including most of South America. So we worry about bird flu, but I would argue we may want to be more worried about pig flu. Because of what's happening in the state fairs all around the US, where we have kids, and this is literally a picture from a state fair where they're in sleeping with their prize pigs before they go show them, and people with their babies in strollers right by the pigs. But there are viruses jumping from the pigs into people that are H1, H3, but they're different, right? So these HAs and NAs have changed in such a way that our vaccines and our previous exposures will not protect against these. So we think about reassortment in pigs, in people, but what about if it can just adapt? What if these viruses get in and start adapting? And that's where antigenic drift becomes important. So antigenic drift is basically the virus has to bring in its own machinery in order to replicate. And this machinery has no way to correct errors as it's going along and replicating the virus. So think about turning off spell check. And this is actually one that I typed up just because, and you can see how horrible it would be if I turned off spell check. But that's the same thing with the viruses. It makes these point mutations. And for the most part, these viruses are defective. They're not going to replicate. But there are cases where they can. And those will be the ones that will allow you to bind to new receptors, evade your immune responses, potentially get into new species. And this is why we have to update our annual flu shot so frequently. And we've started thinking about this adaptation and this antigenic drift. And is it possible that there are reservoirs for this as well? Are there people that might support or allow viruses to emerge? And as you've heard, we've been looking at the impact of obesity on influenza virus. So why obesity? Because during the 2009 pandemic, for the first time, overweight and obese people were shown to be at a very high risk for developing severe influenza infection. And this continues to be true with seasonal influenza virus. Increased hospitalizations, time in the ICU, and even mortality. Now, why is that? Because when you're overweight or obese, you have this chronic low-level inflammation that doesn't allow your immune cells to function properly. So basically, you're in an immunocompromised state. And what made us start thinking about why this population may sort of help virus drift a little bit differently is because of a few papers that came out. One is that in college students, this was a study at the University of Maryland in college students, and what they found is that college students that were overweight or obese actually exhaled more virus in their breath. And the other thing is a study from Nicaragua in a household transmission showing that overweight or obese adults actually shed virus longer. And if they're asymptomatic, they can shed it twice as long. So we know from animal models, we know from this, the virus has more time to replicate. It's shedding longer and potentially may drive more drift. I guess it's important to note, we also know that this population is more likely to get infected with the virus. And what we found in animal models is that when an obese animal gets infected with flu, those first epithelial cells in your nose or in your lungs actually don't recognize that virus. They're delayed. They don't see it. That SOS alarm doesn't go off for about 8 to 12 hours when it should go off immediately. And what this is allowing is the emergence of new little variant viruses because there's not as much pressure on the virus not to change. Does it happen in people? What we can say is if we isolate nasal or lung epithelial cells from overweight or obese people, the virus replicates faster, and it is linear with the body mass index, and that these viruses that are coming out faster and at higher titers have many more changes within that population of virus. So we're now actually looking in people, both children and adults, in viruses from these populations. and looking at whether we get increased antiviral resistance. Now, this is not just this population. We think this change or this problem with the cells not recognizing the virus happens in most high-risk populations. We're looking at pregnant women as well as the very young and the very old. So we've got a lot of flu out there. We've got changes in our host populations which may impact how the virus is drifting. What are we going to do about it? How are we going to know what to be concerned about? Well, I think the first thing we really have to do is we need to be out there looking for influenza virus in people, in the animals, and most likely at this animal-human interface if we're worried about emergence. We need to know what's out there, where is it, and how is it behaving? Is it transmitting? Can it jump species? You know, and these are difficult studies to do for a lot of reasons. The human studies are easy. The animal studies are difficult, especially if you're talking about looking in birds. The reason for this is that if we find influenza in birds, especially in poultry, and it happens to be certain strains, it's reportable. You have to report it to the government, and it actually has significant economic impact on that country where they can undergo trade embargoes. And so it's something that many countries don't want us to come in and actually look in their populations. And there's also a lot of social stigma associated with those people that might have animals that have influenza virus. You can do active surveillance. You can look for disease. And again, we have to do this in the people. This is going to be critical for our universal vaccine efforts because we have to know what's coming down the pipeline in terms of viruses to make sure that our vaccines are continually protecting against the viruses we don't even know are coming yet. So if it's disease, that's pretty easy. You can do severe acute respiratory surveillance. You can go in hospitals or in outbreaks. But what if these people or these animals have no disease, right? If somebody doesn't have any disease, you're less likely to volunteer to go be part of, we're going to see if you have influenza virus. And does this really happen? It does. We heard a little bit earlier about Native Americans being amongst our highest or most vulnerable populations. And we actually have a cohort of indigenous people in South America that we're studying with the hypothesis being that they would have been the most vulnerable population. They don't get vaccinated. And so we asked, are they getting influenza virus? And if they do, is it coming from the animals or the humans? We've been doing this for 4 years now. We have not found one person, because we're doing fever clinics, with clinical disease. And this is some of our populations with their pigs and their chickens and the huts that they live in. They live in those huts with their animals. Not one person has had clinical disease. Most of them are seroconverting. So that means what am I trying to say? I just lost my word. But they're being exposed to human viruses and they're being exposed to animal viruses. And we hypothesize maybe these animal viruses, constant exposure is giving them some protection, but we don't know. We can also look at antibody conversion, and this is what we're having to do with our indigenous cohorts. And all of this together is what's going to be important for understanding sort of the epidemiology and the emergence of influenza virus. But what about when we find viruses? How do we know? We find a lot of viruses all the time. How do we know which ones might be a risk? Well, both the World Health Organization and the CDC have come up with algorithms that we can use, and we can do a number of laboratory-based studies to actually predict or help predict which of these viruses may pose a risk. We can look at things like the properties of the virus, including what receptors does it bind to, really important, does it transmit, attributes of the population, are these people immune to it? Would you be immune to it if it was to emerge? And then looking at the distribution. And all of these together, we can then use that algorithm to actually make some predictions. And this is an old prediction, but you can see the impact risk versus the emergence risk. And we can use that to actually prioritize the funds, which are limited for things like this, in order to determine which ones should we be most concerned about. And the key to this is going to be working together, both the people doing surveillance, the basic laboratory people, in order to really be able to better understand influenza virus and ultimately make a universal flu vaccine, which will be the topic of the next speaker. And I was right on time. So with that, happy to answer any questions.
Thank you. So let's have 2 questions for Stacy.
Nancy Moses from Philadelphia. So Thanksgiving's coming, right, Bert? And we have an intergenerational table with some young people, some older people, and some people who are considerably larger than other people. Should we rethink our Thanksgiving dinner guest list?
I guess that depends on if you would like to rethink that and use this as an excuse, but absolutely not. You know, that's going to be the important thing is we know the vaccines are not as effective in this population, so I think what that argues is it's more important than ever to make better vaccines, but also to make sure that everybody else is vaccinated to protect those that perhaps can't get vaccinated.
Joel Cohen, Rockefeller University, New York. Thank you for your talk. Could you address the question of causality in the association between obesity and viral exhalation, viral load, viral duration? Do we know whether the virus is causing the obesity, is a consequence of it, or is merely associated with it? What is going on causally, please?
Well, I don't think anybody would say that influenza causes obesity. In terms of what's happening with the disease severity and why the higher exhaled breath, what we can see is that the virus is replicating faster into higher titers, so it would make sense that you would get more virus actually being exhaled. Transmitted, we don't know. We're looking at that yet now to see if it does impact the ability of that virus to transmit better, since it is being shed longer and at higher titers. We would predict yes, but we actually need to test that.
Well, thank you, Stacy.